Okay, so we're going to continue to show what happened before and what happened next to try and provide a little continuity. So last week, David gave a lecture on descriptive statistics, things like mean, median, distributions, visualizing box plots and other ways of visualizing data. And that theme will continue, but we're going to start mixing in some other themes at the same time. Today, I'm going to talk about data types and representations in molecular biology. So specifically, types of data that we use in high-throughput genomics and how that relates, not so much to distributions, but to having sort of a computational approach to the data rather than a one-by-one, do-it-by-hand kind of an approach. We're also going to be mixing in some biostatistics work, and there's going to be a lecture on probability in 2 more days. Okay, so in this lecture, we're going to talk about text formats for some common genomics data types, formatting data, specifically standard formats for particular types of data, some basic database concepts that are good to have in mind. We're not going to be studying database languages, but it's good to understand how databases are structured and how you can interact with them to get the data that you want. And then we're going to have a little tour of some of the most popular molecular biology genomics databases. GenBank, dbSNP, GenBank, genome browsers. And we're also going to talk a little bit about how to structure database queries to get what you want. So all of this is about biological data, and I like to simplify. Biologists collect lots of data. There's hundreds of thousands of species, millions of articles in the scientific journals, and then there's genetic information, gene names, phenotypes, locations of genes, mutations, and other landmarks on chromosomes. There's linkage. There's all types of data that tend to be reported in large batches in papers and then stored in public data sets, databases. And it's particularly important that you be able to access those data, compare to new data that you're generating experimentally. High-throughput lab technology has greatly increased the data production from each investigator, each project. You've got multiplex PCRs, gene expression microarrays, rapid DNA sequencing, many other methods of collecting genotype data. And it's worth thinking about data quality assessment, although that's not the purpose of today's lecture. That kind of goes back to what David was talking about last week, variants, etc. So there's a lot of different technology that's showing up in our labs these days, automated ways of collecting data. The raw data from those machines might be stored in a machine-specific custom format, an image, a trace file, or just some binary data format that the manufacturer of that machine finds convenient. In order to publish scientific data, it's generally converted to a standard text format. And why do we publish scientific data and store data in text? It doesn't require custom software to read the data. It's stable for long periods of time across different computing systems. So the ASCII format is universal. You can send an ASCII file from Mac to Linux to Windows. You can access data that was deposited in GenBank 20, 30, 40 years ago. You can't read data off a 30, 40-year-old computer very easily. The disks are different, the file formats are different, etc. But the text itself is an enduring format. And it can be smoothly shared across many different computer systems. The Internet, World Wide Web, is basically a text-based medium. Text itself isn't a format so much as a standard. And within text, you can have many different file formats. We're going to talk about FASTA especially today because it's one of the most important file formats for the exchange. Well, it is the most important format for the exchange of sequence data, both DNA and protein. It's an enduring standard. It's been around for a long time, and all software that deals with sequences use it. We're also going to briefly look at FASTQ, although I have another lecture on next-gen sequencing that's going to go much more deeply into it. But that's a modification of the FASTA format that allows the sequencing machine to store quality information about each base in the same file. But a program that was designed to read FASTA data isn't going to know how to read FASTQ data. It's a completely different format with its own standards. I'm also going to look at things like GFF and GTF and a few other file formats just to show the sort of breadth and expandability and flexibility of these fairly simple text formats. I'm also going to talk about a data storage paradigm that I call tag value pairs, but you could think of it in a lot of different ways. But you've all seen databases where there are a series of fields: the address, the phone number, the username, the user ID, and each one of those fields has some information in it. Every record, every individual in the data set has all of those fields, although you don't necessarily have information in every one of them. You could take all that data and print it out as one long text file as long as you have something that separates each record. So you just have the tags and you have the values. And I'm going to show you a couple different ways that we format those in text. HTML is another kind of a tag value pair where the tags are like these things that we call HTML tags, like bracket B for bold and close bold, bracket A href for a link, etc. And those are not ordered, right? You can, in an HTML file, you can have any number of tags in any order you want, but still it formats the text in a specific way. There's a similar standard called XML, where instead of using a known set of tags like bold and href and style, you can create your own tags at the head of the document and then use them later on. And the GFF is a little bit more like that. You can define some of your own fields early on in the header and then use them later. It has the downside that different GFFs coming out of different systems don't look like each other because they've defined a different set of tags. All right, to talk about what is a database, and I have a couple of metaphors, one being the kitchen recipe file box or the address file folder, and the other being an Excel spreadsheet. And the important thing is to understand how these things are the same. And in the index card box, each index card is a record, and it contains information about one particular thing. If it's a filofax and those are address cards, then there's a name, address, phone number, email. Those are fields. If it's an Excel file, then the name, address, email are going to be your columns, and then the rows are going to be the individual people. If it was a recipe box, then you might have a list of ingredients, and those would be your columns, but that wouldn't make as nice a database. So, record is an individual item that has information in different fields. A record is like a submission to GenBank, a particular sequence record. Or it's an individual in your file card box. A field is a category, and it goes down. Every record has a space for that field. You may not have any information there, but you still have that cell in the spreadsheet. So records contain data that's related to one object. A record does not need to have data in every field. And you could also think about this going back to the previous metaphor that it's a tag value, right? The tag is the name, the SNP ID, the value is what that ID is. The gene name, the description, header, whatever the description text might be. You can treat a spreadsheet as a database in the sense that you can search in one column for a particular item rather than search all of the text of the spreadsheet. And that's going to be pretty valuable. If you're looking for a particular word in the gene name, that's going to be much more helpful than searching everywhere, because that word may turn up in a different context somewhere else in the data record, in the description field. Or in this case, when we're talking about genotypes, you want all the C's in the first haplotype, and that's quite different from a C in one of the other genes that are also in this record. So you could sort and search, or you could even search for recombination. You could search for a C in hap A and an A in haplotype C, and that will give you a specific set of records. So the fact that the data is organized into fields, and you can query specific fields, and then you can build intersections between those fields, field A and field B type queries. It gives you a great deal more power to slice and dice the data. Oh, that's weird. I guess I'm having Mac problems. Sorry. I may have to come back to that page after I've done it again. So I'm showing an Excel spreadsheet here, and then on the right and the left, I'm supposed to be showing how you could save that as a tab delimited file, which is just a bunch of text, or a comma delimited file, which is also just a bunch of text. I'll have to remake that slide. Give it back to you later. Oh, there we go. Tab delimited. Huh. I can take it all back. All right, so this thing could be saved just as a text file, right? And the tabs are saved, and then the carriage returns are saved. And if you don't open it up in the right software, it can be kind of a mush of data, but it can still be read. Important thing is that if you lack a value in one of these fields, you still have to put a tab there or a placeholder of some kind, because the organization, the number of tabs before you hit the carriage return, is really important. Same thing for this bottom one. Same data, but now it's separated by commas instead of tabs. And again, if you leave out a value, you need to put a placeholder there in order for it all to fit together. Okay, so to talk about standard formats, it should be pretty obvious that the same sort of data generated by different labs or different machines ought to be in the same format. Surprisingly, that's not always the case. In new technologies, there tend to be more than one version or more than one vendor, more than one company who's selling a machine, and they each have their own format that they prefer for whatever reason. And there tends to be a few different competing formats. Sometimes a standard wins out. Sometimes there's an international body that gets together and says this is a standard. Other times it just wins by force of numbers. In sequencing, the FASTQ format won out because Illumina became the most popular company for sequencing, and they supported the FASTQ format. And the format that was preferred by 454 disappeared, essentially, and no one is really posting data to GenBank in that custom format anymore. So sometimes these things just evolve, and other times we have an actual meeting of scientists to establish an official standard for a particular data type. Other times, a very influential database will say, this is our standard, and that will become universally adopted, or at least so common that everyone also puts their data into that file format. So to talk about a standard for DNA sequences, I've already said that text has a lot of power in terms of being cross-platform and enduring over time, but just saying that DNA should be text doesn't answer all of the questions. How many letters should you have per line? How should you handle spacers? How should you handle unknown bases? How should we distinguish the header from the sequence, and what should go in that header? A lot of sort of unanswered question. And then, should we have a completely different file format to store protein sequences, or can we mush them both into the same format? So William Pearson developed this format called FASTA for a piece of software that he called FASTA, which just stands for Fast Alignment. It was one of the earlier alignment programs that worked pretty well at scale. You could align hundreds of sequences in a few minutes on computers of the 80s. So Pearson's format was made strictly for his own purposes. He had developed some software, and that software needed to read data in a consistent file format. And so this was what he came up with. There's a single header line that starts with a greater than symbol, followed by free-form text. By default, there's no space following the greater than symbol, and the first word, meaning all the characters until you come to a space, is defined as the sequence identifier. And also, kind of by default, that sequence identifier is unique. So in your data set, every sequence should have a word following the greater than symbol that uniquely identifies it. And all databases follow that rule. And in addition, all public databases respect each other and don't create different sequences with the same identifier. So if there's a GenBank ID, it's not going to be used to identify a completely different sequence in a different database. Following the first space, there's no specifications in the official FASTA format. And in fact, every piece of software and every database uses the rest of that header line in a different way. And you can develop your own software to parse the file format of a FASTA file that comes from a particular source, particular database that you like to work with, but there's no certainty. In fact, there's almost no chance that that same header will be formatted the same in a data file from a different database. Even if you get the same exact sequence from GenBank and EMBL, the header is going to be formatted completely differently. And in fact, the sequence identifier is going to be different. EMBL is going to have an EMBL ID, and then the GenBank ID is going to be embedded somewhere else. GenBank is going to have its unique identifier as a GenBank ID, and the EMBL ID may or may not be embedded somewhere else in the header. The header goes as long as it needs to go until it hits a carriage return. So if you see multiple lines of header, that's really wrapping, and there's no carriage return in there. That's part of the definition. From the greater than sign till the first carriage return is the header. After the first carriage return is the sequence itself. And the sequence is not specified by FASTA. Usually you need to know whether you're dealing with DNA or protein sequence. The DNA alphabet is further defined as GATC and N, but there's no rules about whether there should be spaces, dashes, dots, or numbers, or other unknown characters in there. Your particular software may fail because some characters have been embedded in there by somebody else's software writing to a FASTA format. So this is a place where the standard ought to be stricter. GenBank has a stricter standard, Ensemble has a stricter standard, but some software that you're working with can export FASTA file and embed characters that some other software won't like. Had that happen yesterday. Frustrating. I think that's all I need to say about FASTA. Oh, multiple FASTA. You can have multiple sequences in a single file and still call it FASTA. At the end of one sequence, there's a carriage return. Then you start a new line with a greater than symbol and a header. Next carriage return is the next sequence, followed by another carriage return, greater than symbol, header, and as many as you need. So you can get a dump of an entire genome or even an entire section of GenBank as a single FASTA file. It could be many gigabytes in size. Obviously, it would be useful to have software that could pull out individual sequences from this big file according to their unique identifier, and that's actually the exercise we're going to work on today. If you look carefully, this particular FASTA file comes from Flybase, which is a Drosophila genome database, and they make really heavy use of the header. If you look carefully, there are a lot of key value pairs, and they use an equal sign to distinguish what's a key and what's a value. So they have a type equals protein, and then they separate the key values with semicolons. This is just their personal convention for how to write the header of a FASTA file, and they're compressing a lot of Flybase information in there. Because they include the keys as well as the values, they can put them in sort of arbitrary sequence, and they could leave out keys that are irrelevant. They don't have to put a dot for every field that has no information. So that's yet another way to use the key values. So it's a little bit more like the HTML model, where you have a tag and a value, but it can be located anywhere in the string, anywhere in the sequence. And then, like I said, the long header just wraps until it comes to a logical ending point, and there's a carriage return, and then there's the sequence. These are protein sequences. They seem to be formatted with some tabs and spaces, and that stuff doesn't matter. These are valid FASTA files, or this is a valid FASTA file. So you could, in fact, write some software to look for chromosome locus or parent or cross-reference to a Flybase ID in the header of each of these. But that same software wouldn't work on data from, say, C. elegans database, because they choose to format their headers differently. Okay, so that's FASTA. That gives us a standard for DNA sequence and protein sequence data, but obviously there's lots of other molecular biology data that we would like to exchange in common standard formats. Do we have standard universal standards for all these data types? Genotypes and haplotypes, clinical records, gene expression, genome annotation, protein structures, alignments, phylogenetic trees. And each one of these is like a big topic and an essay in and of itself with a lot of history of science built into it. So in some cases, standards have evolved and come to dominate. For example, protein structure uses the PDB format because there's just one very well-used database for the whole protein structure community. All the software reads PDB format. And if you're going to publish a structure, you're definitely going to publish it in a PDB format. Clinical records is the worst. Every hospital and every doctor's office has a completely different clinical record system, and they're completely incompatible. We just spent hundreds of millions installing the EPIC database here, and I've come to learn that an NYU EPIC file is incompatible with an EPIC file at any other hospital anywhere else. So that was a really brilliant effort to maintain the incompatibility of clinical records. The best that we have, I think, is a database at NCBI called dbGAP, where they have some standards for clinical phenotype information associated with genetic studies. But it's a tiny little sliver of the clinical data that physicians can possibly collect. And in fact, each dbGaP study has its own unique fields that they define for themselves. So you could have two studies on the same disease that have fields defined in different ways and have essentially no way to automatically compare them. And other things in here are fall somewhere on that spectrum. Phylogenetics is a pretty good example where there's 2 or 3 very common formats that most software can read, some of, or most of, or all 3 of them. And there's a dozen other less common formats that a particular software author has decided they need to create yet another phylogenetics format to support the special features of their software. And if that software should become popular, then somebody else may read and write that format as well. Alignments is moving a little more in the standardized direction because there is one really popular software called Clustal that generates alignments, but there's 6 other really common alignment formats as well, including a FASTA format that just has spaces mixed in with the text that allows you to line things up, but not every piece of software respects those spaces and allows you to redraw the alignment if you save your file that way. So it's a messy universe, and as soon as you start working with a particular data type, you have to ask yourself the question, what is the standard file format for this data? Who's publishing the data? Where can I get it? What are the common databases? Am I going to have a problem converting from the database that has the database I want into the software that has the function that I need? And that ends up taking a way large chunk of bioinformaticians' time, is doing those file format conversions. So we will revisit that probably a few more times in the course. Here's an area that's really bad and really ought to be better because it's an important field. Genotypes. There are a lot of studies in the area of GWAS, which is genome-wide association. There's several competing technologies from several different companies that can assay up to millions of different individual polymorphisms, specific single bases on the genome, and report whether you have a G or whether you have an A. But since true genotypes are slightly more complex, you can be homozygous or heterozygous, and the software can read that out. There are not 2, but 24 formats for reporting SNP results, and whether you're heterozygous or homozygous, and a whole wealth of software that has some ability to convert from one format to another. So if you're going to work with SNPs, yes, you could work with the software that was developed by the manufacturer of the machine that generated your raw data. Otherwise, you're going to be in some world of pain trying to find a conversion method to get the data out of that format and into the format of the tool that you love best. So I said this, messing around with the results, creating summaries, etc. So having some fluidity with software that can read text, tab formatted text, and swap between the columns, name those columns, and use them later is going to be really helpful in the day-to-day work of doing data analysis in bioinformatics. So that's the basic message there. So just to emphasize the importance of accessing data from public databases where we have complete genome sequences, SNPs, genotypes, etc., your experimental results are greatly dependent on your ability to access public resources. You know, you're sequencing a genome. You want to know what are the mutations. Mutations compared to what? Compared to a reference genome. You've just done an experiment looking at gene expression. and you get a pattern of genes that are up and down. What other experiments in similar systems have seen similar changes? Or how does this gene that I see as differentially expressed perform in other experiments? So being able to access these public databases is essential for you to understand the meaning of your own experimental work. We're going to start with GenBank because that's kind of the Rosetta Stone of bioinformatics work. It contains all the DNA and protein sequences described in the scientific literature or collected in publicly funded research. That's important to realize that if you want to publish in any journal article, you have to put your data in GenBank. So if anyone has published, then that data is in GenBank. So you don't have to go all over the internet and to people's private websites looking for sequence data. GenBank is a superset of all published sequence data. Not all kinds of data, but when you're talking about sequences and you're talking about being published in refereed journals, there's a universal requirement to submit that data to GenBank. As I mentioned before, text is a universal format, and you can get data out of GenBank in what they call a flat file, which is simply a formatted text file. And I'll show you an example of that in a second. Every submitted sequence is a record. Sequences can be really small, like a primer or something like that, or it could be really large, like an entire chromosome from a de novo assembly project, or the complete genome of a bacteria. That can be a sequence record. Each has fields for organism, date, author, etc., and each one has a unique identifier, usually called the locus or an accession number. Those are two different unique identifiers that every GenBank sequence has. So here's an example of a GenBank record for the yeast TCP1 beta gene. And here's my pointer. So there's a locus name for this thing, SCU4985. There's an accession, U4985. And there's also a GID, which is an internal GenBank identifier, not generally used by other databases. So it's a G1293613. All three of those identifiers are guaranteed to be unique and to point to this sequence record. The locus name won't change. If the sequence is updated, the locus name will stay the same. The GID will probably change, at least incrementally, to a .1 or to a .2, .3, etc. Then there's all these other named fields, and some of them, a lot of them are required. You've got to have an organism for every sequence. You probably have to have a reference with an author and a title and a journal, but not necessarily. You can have that field automatic submission, meaning there's no published journal article about this sequence. We're just dumping a bunch of sequence because we want to, and GenBank will take that. There is, however, a person who owns this sequence, which is the author of the sequence, not of the reference. And then there is a feature field, which is sort of an unlimited area where you can have many, many different features attached to a given sequence, as many as you feel like adding if you're the person submitting the sequence to GenBank. Some of them are standard, such as if there's a coding sequence, if there's introns and exons, but you can annotate a feature, say that there's a known PFAM domain or a known domain that is bound by a microRNA or a known upstream region if you wanted. Anything you want. There's a really large menu of features, and then there are there's also a domain where you can put unspecified feature, just like an extra feature, and then just a note that you want to put something associated with this sequence or a particular location on this sequence. So it's highly structured in terms of the field names, but then there's also some space for a little bit of innovative work. Yeah?
What kind of checks and balances are there for these records?
It's a two-stage process. There's an automated submission tool where you have to fill out all the required fields. And it also checks, there's a translation field if you're submitting a DNA sequence, and your translation has to match the translation that they compute based on the start codon that you specify. Right, so you're saying that the protein coding sequence starts here, they'll translate it and check that the translation that you put matches. So there's a few automated checks. And then it goes to a curation step. Now, they don't have an infinite number of curators. So if you're just submitting one sequence and millions of other people are also submitting just one sequence, it's probably going to be fine. But people don't tend to submit just one sequence. You tend to submit a batch. And then that whole batch goes through a curation process. And that curator will get back to you with comments or problems, and then you fix those comments or problems, and then your sequences are finally loaded in. I've been through this a few times. So there's an automated phase and a human phase.
But if you're searching for a record and you want to find the plasma membrane glycoprotein, how sure can you be that this is actually a plasma membrane glycoprotein? Because they've said so.
Because there's an author for every sequence, and usually there's a journal reference as well. So ultimately, the validity of the sequence rests on the reputation of the author, which is further bolstered by the citation of the reference. So you don't tend to do it, I mean, you can go to GenBank, put plasma membrane glycoprotein in the query field, and find the things that are called that. The other way around might be more sensible, to first search for an authoritative journal article, see the GenBank ID of the sequence that they've submitted, search the database using that unique identifier. Now you know you have the sequence that they were talking about in that article. Now somebody else may have submitted a different sequence for that same gene, and now you have to make a decision yourself as to which one you prefer to use. And hopefully they both have journal articles associated with them, and you could read about why they may be different. So the database just sort of is what it is. There's another layer called the RefSeq, which I'm not dealing with right now, where for a particular gene in a particular organism, a curator has chosen one gene to be the reference sequence for that gene. So you could go and say, take the human RefSeq, and there's only going to be one human RefSeq for plasma membrane glycoprotein alpha. So that's one additional layer of curation. And not every sequence makes it into being a RefSeq, and not every gene has a RefSeq. I mean, most human genes do, some mouse genes, other organisms very spotty. Well, Drosophila has RefSeqs, but less model organisms may not have a full set. I made that mistake with the pig. There's only 6,000 RefSeqs for pig. So if you choose the RefSeq annotation and you do a gene expression experiment, you're missing 2/3 of the genome. Just as another example of another database is dbSNP, and it's formatted in a somewhat similar way. You've got the tags and the values again: molecule type, genomic, creation, update, date, variation class, allele origin. Some of these headers have no data in them, so it's an empty field. And then there's this FASTA format here, here in the middle of the reference, where there's a header, there's a SNP ID, which is unique, RS1042752. That's up here as well in the header. SNP cluster report. And then interestingly, the way they've chosen to format a SNP, because a SNP is a variant, it shows you the difference between a particular sequence and the reference genome. And so what they've done is they're using a Y symbol, which is a DNA ambiguity symbol, to show that there are two possible sequences here, and this particular SNP is different. What the Y will do in some software will allow you to align a sequence that matches either the reference or the variant to this region. And then what they've done is they've surrounded that variant position with about 50 or 60 bases on either side, which will allow you to search the SNP database by sequence, by doing a BLAST type search. So the question we had was about, how do I know I'm looking at the right sequence, which brings us to this concept of accession numbers. Unique identifiers are the way that databases are designed to be searched. They're not designed to be searched with free text, and the result that you get with a free text search is sort of unpredictable. Particularly, well, genetics is particularly bad in terms of working with text because people will name genes, SMOD-like gene, or not really the SMOD gene, but we thought it was, or something along those lines. So the name of one gene can be embedded in the name of a completely different gene, or it could be, the insulin receptor, right? It's not the insulin gene, it's the insulin receptor. So if you search for insulin, you're going to get both. And then there's probably insulin receptor variants and insulin receptor modifying protein and that sort of thing. Whereas if you go to an authoritative source, such as a journal article, you get the accession number of the actual molecule that they're talking about. You can unambiguously pull out that sequence with that accession number from any database. There's also this idea of linked databases, which I'll get to in a second, where the accession number serves as a key that takes you from DNA to protein to SNP to structure, etc. Had a problem with an article that was published just a couple of years ago. And they used what should have been valid accession numbers, but they all changed. They used XP, which are sort of temporary protein IDs, and they all changed. So the entire conclusion of the paper is unrepeatable because you can't get the sequences that they used. Okay, a little bit back to GenBank. GenBank is managed by the National Center for Biotechnology Information, which is part of the National Library of Medicine, which is part of the NIH. There was a time when you could subscribe to GenBank and they would mail it to you on a stack of CDs. I have some of those. Now GenBank is over 150 billion bases and they no longer print CDs or DVDs or anything else. People access it over the internet. To download the whole thing, which we also used to do around here, is not something that anyone does very much anymore, although there might be reasons to download a particular section. We did all the bacterial sequences a couple of weeks ago because we wanted to do a local search for bacterial contamination in our genome, and it wasn't feasible to do that all on the NCBI side. Right, so we always show this nice graph, the growth of GenBank, and you'll notice how it gets steeper as we go on in time. There's not too many exponential growth curves where the angle of the exponent keeps increasing. So you'll also see that the doubling time of GenBank was traditionally about 1.5, 1.2 years, and now it's squeezing shorter. And that's strictly a result of improved DNA sequencing technologies. And people are dumping more data faster into GenBank. And that trend itself is likely to continue, because there's a lot of research now that says we don't need a 1,000 genome project, we need a 10 million genome project to get enough background information on the frequency of variant alleles that we see sometimes. You know, are these variants really pathogenic, or are they just rare? So it's cheap, and there's scientific reasons why we need more data. We're also sequencing more genomes, strains of pathogens, new organisms, and there doesn't seem to be an end in sight. All right, I'm also going to talk a little bit about relational databases. Up till now, I've been talking about a database where the metaphor is simply an Excel spreadsheet where you've got your rows and you've got your columns, your fields and records. But if we think about a situation like a set of protein sequences in GenBank and a set of DNA sequences in GenBank, even from the same organism, but they could be from any organism. Some of the fields that you would use to describe a protein are not relevant for DNA, right? Proteins have active sites, proteins have all kinds of features. And DNA also has features such as introns, such as untranslated regions, that's not relevant for a protein site. So if you had one database, fields and records, you'd have fields that would be empty for all of the protein sequences. And that would make for a messy and unbalanced sort of database. And other fields that would be empty for all the DNA sequences. Yet we do want to maintain some sort of a relationship between the protein and the DNA for the same gene. And that's where we come to the concept of a relational database. And the easiest metaphor for that is a stack of Excel spreadsheets that are linked by one particular key value. So there would be a particular value, a unique identifier of the DNA sequence, which would show up in a particular field in the protein sequence, and vice versa. There would be a unique identifier of the protein that would show up in a particular and predictable field in the DNA record. And that link would allow you to establish a relationship between the two. And there may also be other fields that are shared. They could be submitted by the same submitter at the same time. Some of those fields could be shared, but they don't have to be. Okay, so those linked spreadsheets are called tables. And the whole structure of interlinked tables is called a relational database. So GenBank also has a relational database as well as this flat file structure. It's not really feasible to print out the text of a relational database. You can only really print out the text as a flat file of one table. The relational structure just doesn't really translate into text. There are many different tables in this vast interlinked database that is hosted by the NCBI. For example, there's all of the published journal articles known as PubMed. There's a set of genetic diseases, OMIM, the polymorphisms in dbSNP, cytogenetics, gene expression, taxonomy, chemistry, etc. You can only do you can do a query across different tables, but it's more complex. Generally, databases are designed to allow queries on one table, and you can do Boolean queries, meaning joining a question about one field with a question about another. So you can search organism and gene name, for example. And those are fields in one table. It becomes quite challenging in database theory to search across different tables and have a linked search, so I'm not going to talk about that. Just in general, you want to search the most specific place possible. So if you're looking for human sequences, it'd be much better to search for Homo sapiens in the organism field rather than in the general description field for human, because you never know what might come up. You might get human immunodeficiency virus, which is a valid search result, but it's not really a human sequence. Also, in terms of internet, we very frequently have a nice, friendly, easy query interface that does the simple thing. It searches some combination of the most frequently searched fields, and it just has this one little place where you can type and hit one button and do a search. And that's the Google metaphor. But in fact, Google isn't such a simple thing. It has fields and databases, and there is an advanced query page where you can specify queries limited by country, by language, by date of publication, all these other things. File formats, you can look for only PDF files, you could look for only images, et cetera, et cetera, et cetera. And so there's more going on behind the scenes if you just go past that very first friendly page. GenBank is the same way. When you come into GenBank, there's one field. It always looks different. I haven't tried to update my picture because they change their front page really, really frequently. But it always has this one query field, with the caveat that there's a pull-down menu to the side of it that specifies which of the many interlinked relational tables you can search. There is, like in Google, an all fields search, but you probably don't want to use that because it will give you many hits across many different databases, and then you'll have to spend a fair bit of time sorting them out. So you should have a basic idea in your head whether you're looking for a DNA sequence, a protein, structure, taxonomy, journal article, etc. So, let the GenBank interface, the Entree interface, know what you're looking for, and you'll get there a lot faster. There's also these advanced query interface where you can specify. Now, realize that GenBank is part of the National Library of Medicine. And so a lot of their tools are designed from a librarian's point of view. So key things about a GenBank record from their point of view is the author. Whereas that might not be so important to you if you're looking for, an alpha cation channel from yeast. But organism is a searchable field. You could search just for the RefSeq, which would mean the reference genome. You can search, I guess nursing jobs isn't really that relevant, but there are a lot of fields here that you could specify and send your search to. And there's also a query builder where you can create Boolean queries, this and this or that, but not this. So the advanced search tool will get you where you want to go if you give it a little time. The Entree database is also interesting in that it has pre-computed links between some of the tables. In other words, they have anticipated some of the things that you might want to do and have done them in advance. For example, among the DNA sequences, they have already done internal blast searches. So for every sequence, there's a set of links on the right to all of the high E-value or strong E-value blast matches. For many sequences, there's a link between a DNA sequence and the protein. For many proteins, there's a link back to the DNA and also a link to a structure. There may be a link if it contains a conserved domain, etc. So they describe this like this, but it's actually a fair bit more complex. But the idea holds that for every nucleotide sequence, there's probably a journal reference in Medline. There's probably a protein. There's definitely a genome, although not necessarily a complete genome for every DNA sequence. There may be a structure, but structures are really linked to proteins more than they are to DNA. So this isn't even the whole thing, but this is sort of a picture of how they conceptualize the whole interlinked structure. Interestingly, the thing that has the most links are the journal articles, which, if you think about how science is structured, makes the most sense, right? No matter what type of data you're generating in your work, you're going to want to publish articles about it. And the refereed journals have come to an understanding and published a set of rules that if you're going to publish an article about any type of data that is maintained in a public archive, you have to do a data submission. So for every chemical, for every structure, for every protein, for every DNA sequence, there's likely to be a linked PubMed article. Okay, so you say you do a query. This one is a nucleotide query for this thing, BRI. Frequently, you'll get a long list of matches. You have the choice of sifting through them one by one to see if they are what you want, or going to the advanced query interface and trying to filter down your set of links, say by species, by refseq, rather than just any sequence that anyone has submitted, etc. So some other important databases that you'll make use of: genomes, proteins, biochemical and regulatory pathways, gene expression, genetic variation, protein-protein interactions, and gene ontology. I'm going to visit these really briefly today, and then in future lectures come back to most of them in more detail. So this is more like just a tour, but it's fine if we want to stop for questions. So one more data type that I'm going to introduce in a second is relevant to these genome browsers, which is a relatively new public data resource. The genome browser was only really created in 2000, 2001 to host the Human Genome Project. When they finally reached the point where they could build a consensus genome from all the bits and pieces, Jim Kent at UCSC came up with this idea for a horizontal metaphor. Just lay out the chromosomes one after another and create a search interface that allows you to jump there. That turned out to be a really powerful metaphor, and it allows you to have one of these really easy Google-like search boxes where it takes what you put in and does the logical thing with it. So it sort of first searches for gene names. You can also put a position here. You could put a GenBank ID here. You can put a SwissProt ID here. And it will search successively through these different tables trying to find a match to what you've asked. And if you have an ambiguous query, it may give you a list of possible matches. He also created an interface using a thing called BLAT, which is a fast alignment searcher, and we'll talk about how that works in a couple weeks, where you can paste in a sequence and it will jump to that sequence or something close to it on the genome. It's a lot faster and a lot less sensitive than BLAST and some other alignment tools that we'll talk about, but it gets the job done. So if you have a sequence, say you've generated a cDNA in the laboratory and you want to know what gene it comes from, this is a possible way to just jump right there on the genome and find the coding sequence for that. So here I want to stop and talk about the BED format, which was also invented by Jim Kent in order to support his genome browser. So the BED format in its simplest form is just 3 columns: chromosome, start, and end. And that turns out to be really powerful when you have a genome laid out one chromosome at a time, end to end, because it defines a locus, a region, a position. And that position can be really small, like a single SNP, or it can be pretty big, as big as you want it to be, more or less. A whole chromosome would be the maximum. And it says you could do something with that particular interval, like color it blue, or make it tall, or something along those lines. And it turns out that when you take a set of genome annotations, such as EST sequences, or genes, or proteins, or SNPs, and you lay them out across the genome, and then you lay out some other information, then you can interrelate them because they both match the same position. So the bed format can be a little fancier than just 3 columns. You can have direction, forward and reverse. You can have color. You can have width. You can have height. There's a few more items, but it's a really, really simple format at its core. And when you take bed formatted data from a few different sources, in this case we have genes from GenBank, genes from RefSeq, genes from Ensembl, some mRNAs from GenBank, some ESTs. Somebody's built a consensus. We've also mapped SNPs from dbSNP all onto the same location. It just turns out to be really powerful, and it shows the whole value of genome annotation. Putting things together using this metaphor of gene location or gene intervals, rather than worrying too much about the sequence itself. Although there are properties of the underlying sequence that you could map here, like intervals of GC content, whether there's a repeat, whether there's a large-scale segmental duplication. All of these can be tracks. Other things you could put here are gene expression, epigenetic marks that happen to map to this location. So the general concept of a bed file, really simple and really powerful. Of course, you need to have a reference genome to map it onto. So UCSC only supports about 24 genomes. So the metaphor only works when there's a well-supported reference genome that lots of people have generated this location-specific data for that reference genome. In fact, for the human genome, there are a lot of possible tracks. And there's a interface, if you just sort of scroll down in the genome browser, that lets you turn on or turn off many, many, many different annotation tracks that are contributed by all kinds of different investigators. And so this is another way of publishing your data, is you can create a UCSC genome track, and if you've published in a referee journal, they're pretty flexible about hosting the data. And it doesn't really matter if it's not particularly popular, if there's not lots and lots of people using it, because it can be added down here as an optional track and only turned on when someone wants to turn it on. You flip on a particular track, and then it shows up here as another row. And if you're, as a user, you want to see lots and lots and lots of these tracks, you just get a screen that's full of lots and lots of rows. Because they're just simple intervals that are just drawn as blue or red or orange lines, it's not too heavy a load on your browser to load lots and lots of data associated with a particular genomic interval. You know, it's not really loading the sequences themselves. It's just loading this very simple summary of, at this position, draw a box. Naturally, in Europe, they have the same thing, only different. They have their own genome browser and their own database. The Embl is the European version of GenBank, and Ensembl is the European version of the UCSC genome browser. And there are advantages and disadvantages of the two. Their tracks are not directly compatible. They name their chromosomes differently. And so but the format is fundamentally the same, and they do support the same idea of BED and GFF and some of the other file formats. Different genomes as well. The Ensemble browser is a little more automated. The UCSC has a little bit more human overhead, and so Ensemble has been able to load more newly published and partial genomes than UCSC has. So if you have sort of a semi-model, semi-interesting organism, you may find that Ensemble does a better job for you than UCSC. On the pig genome, I found that to be the case. I'm going to flip through a couple other sort of interesting databases and not delve too deeply into them. There's this thing called SageMap, which is a slightly obsolete gene expression technology, but has this really cool interface where you have the body parts and it shows them in different colors depending on whether the gene is up or down regulated. I just love the interface. There's this microarray database that measures gene expression in a kind of a free-form way. What they have is they have a reference database of genes, and you can have many different reference databases of genes. They're called platforms, and then you can publish your own experiment and reference that platform, and then you don't have to have key pairs for every single value. You can just say, okay, for this gene, I have these values for all of my experiments, and for this gene, I have these values for all of my experiments. And it leads to a pretty color-coded graph like this. And then there's tools that can mine the data in this graph, you know, hierarchical clustering and that sort of thing, to find groups of genes that are expressed in certain samples in a certain way. Really important database of single nucleotide polymorphisms, also known as genetic variation. dbSNP at the NCBI, I already mentioned. They also have a genome browser-ish motif, although for historical reasons, theirs runs vertically rather than horizontally. And there's many different zoom levels displayed all at once. And I find this to be a lot less user-friendly to sort through. But if you're looking for some SNPs, you can load up a bunch of SNPs into a query, and then it will jump you to the genetic locations of those SNPs, and you can figure out what genes they're in, what genes they're near, etc. We're going to do a gene expression and SNP lecture in a few weeks future. Two more types of data that I want to talk about, I'm going to finish up pretty quickly, has to do with gene annotation. And we usually think about gene annotation and function as, you know, what's the protein that this gene makes and what's the biological process that it's involved in. The KEGG database took a fairly simple idea, which was biochemical pathways. And there was like this Boehringer-Mannheim poster that went around in the 70s in classrooms that, you know, showed the citric acid cycle and things branching off of it. And so the people at KEGG took that idea and they said, well, how about if we map all the enzymes onto proteins for the genomes that have been completely sequenced? And then we break them down into subsets. So we'll talk about citric acid, we'll talk about amino acid biosynthesis, we'll talk about cell cycle regulation, and we'll draw a graph that, you know, shows the process. The lines actually, the lines represent enzymes, or the boxes actually on the lines represent enzymes, and those enzymes are sort of vertically linked to a set of different genomes. And so you could say, show me all of the proteins from my favorite genome that map to this pathway. It's more interesting when you do it in reverse. Say, I have a list of proteins. Show me what pathways they map to. And, you know, so you get this kind of thing. Another way of dealing with genes and functions that is a little bit more rigorous is this idea of a genome ontology. Well, this is a KEGG pathway, and it shows you the functions. And here's another way of graphing the same thing, where you just make it a little bit more colorful and pretty, but it still has all those same proteins, and again, you could map them across different genomes. All right, protein-protein interactions, I'm going to skip over that really quickly, just to say that we have collected a lot of biochemical data that deal with those interactions, and you could be co-expressed, they could be bound to each other. There's a lot of different lab technologies, and one of the ways of making a graph is to do a cluster diagram and connect up proteins that are co-expressed or show a protein-protein interaction from a particular data set. And there's a number of databases. This one comes from a database called BIND, bimolecular interactions. But it's the same basic idea of annotating a list of genes, right? So if you have a list of genes and you map them onto this graphic, you could say, oh, I can see that I have genes in interacting clusters. So I must have, modulated that pathway so that those genes are upregulated, or I have isolated them in some way. All right, final concept I want to get across is called genome ontology, which is a more rigorous way of dealing with gene annotation. And genetics historically is bad about naming things because people work on different model systems. So people work on Drosophila, and they have a phenotype, and it looks like something, so they give it a name, Sonic Hedgehog. Somebody else works in C. elegans, and they see a different phenotype, and they give it a different name. And then 10, 15 years later, they realize that those two phenotypes actually come from an orthologous gene. So now how do we name that gene in a third or fourth or fifth organism? And that's where the concept of genome ontology came in. We have a convention, a naming body that sits down and says, okay, how do we define the function of this gene? We're going to use three basic categories: molecular function, biological process, and cellular component. And for every gene, every protein, we're going to give it a name in all three of these domains. And since these things, concepts are not very discrete, we're going to create a hierarchical naming system. So we're going to have a very high-level description of a function like cell cycle or ATPase. Then we're going to have a more specific subfunction and then a more specific subfunction. And the same protein might be mapped in several different functional areas because it might be, both an ATPase and a cation channel, something along those lines. So they have this idea of what they call an acyclic graph, which is basically just this hierarchical nested description of how a particular protein has been labeled. So how do we label all the proteins from all the organisms in the world? Well, there's good experimental data on just a few model systems, Drosophila being particularly strong in this area. So where there is experimental data for gene function, a knockout or mutant or something along those lines, that protein will receive its full gene ontology naming system. Then by homology, we will identify the orthologous, meaning the best reciprocal matching protein in other organisms. And there's a little bit of a diminishing return as you get farther and farther away from your reference organism, which is why sometimes genes are named ATPase-like, as opposed to just saying it's ATPase beta 1 or whatever. That its function hasn't been specifically defined in this organism, but it's defined by homology to a reference organism where we know the function. The nice thing about this naming system is, again, if you have a list of experimentally derived genes or proteins, say a gene expression experiment, now you can take that list, map them to the functions, and see if that list is enriched for particular functions. And we'll talk about some software to do that a little later on. All right, yes?
But those names are mostly used for organisms that have a reference?
Well, the names are defined in organisms with a reference, but many ref-seqs, in other words, once you've got a protein that's reasonably well defined for your organism, regardless of whether there's a fully sequenced genome for that organism, by homology to a known organism, such as Drosophila or C. elegans, or human for that matter, because we've got pretty good idea about gene functions for a lot of human genes. You could apply those functions by orthology to your protein. And for some proteins, you can just look up their genome ontology. For a lot of others, you have to do the assigning yourself based on a BLAST search or something like that. And there's a software called BLAST2Go, which is very popular among people who do new genome sequencing to say, okay, we're going to guess the functions of all these genes based on their closest matches to genes that already have assigned functions. And that's the way to go if you're working with a new organism. All right, summing up. Database search strategies. You want to search by accession numbers, unique identifiers whenever possible, and not rely on the general text-based searching of that sort of summary query engine that a lot of databases put up. You don't always know what you're going to get. Start broad and narrow by using the advanced search to make a Boolean query across several fields, limit by organism, limit by ref-seq, etc. If you have to work with names, be aware that your search is neither comprehensive nor all accurate, so you may need to try variants. Search relevant databases, be persistent. So here's the general paradigm that we will revisit in bioinformatics. You find data, download, reformat, run some analysis software, collect and sort, publish. For your own data, collect the samples in the lab, run your analysis, filter the data, put it into a format that your software likes, run the analysis, publish so somebody else can download and do the same thing. All right, summary. Text formats for some common genomics data types, formatting text with tag value pairs, and know how to recognize that when your data contains named fields. Fields, records, tab and CSV, relational formats. A lot of time on FASTA format because I think it's really important, and today's exercise is all about FASTA format. Molecular biology databases, GenBank, genome ontology. Okay, and next week, or Thursday's lecture is going to be about probability. Okay, so if we go back to the website for the course, we have a homework assignment for today, and I think, not sure, me and Pam are going to trade off on who's going to run this.